Hi.
Hi, Uma.
Hi.
Hi, Uma.
Walter and, oh,
Hi.
are they there? Not yet.
Not yet, not yet.
Okay.
Oof. I had to do faculty meeting today and everybody was, are we live? Sorry.
Yes, we are.
Yeah, yeah. Yeah.
Don't chat. Hi, Vijay.
Hello, everybody. Just wanted to say hello from Dresden.
Hi, Martin.
Thanks for having us
Hi.
for the Humboldt Day. It's great.
Yeah.
Such a great
Thanks for
event.
joining.
Thank you for organizing it.
Thanks for joining.
Yeah. Nice to see you,
It's a
Martin.
pleasure.
After a long,
Yeah.
after
Great
a
to
while.
see you
Yeah.
again. Yeah.
Yeah.
Everything
Great.
fine?
Yes, yes. As well as it can be given
Yeah,
the
fine.
circumstances. Yeah.
Yeah. Given these difficult days, yeah.
Oh, it's earlier in the day, so it's still light.
Yeah.
So I noticed that there are some raised
Hi, Walter.
hands.
Oh, in the participants?
Yeah. I suppose you could use the question and answer box for questions. If
Yeah. If there's questions before we start, then that's... Hi, Walter. Hope you're doing well.
Good. Yeah. Thank you. How are you?
Good, good. Hi, Karthik.
Hello, Karthik.
Hi, Uma. Hi, Robin. Hi, Walter. Vivek.
Hi
I
there.
Hi.
So I
Yeah,
was
like-
checking in yesterday. It looks like a successful event. A lot of people participating. So congratulations.
Yeah.
Thank you.
It's been quite fun.
Thank you.
A lot of students interested. So, yeah.
Okay, so I'm going to go live and start.
So Vivek, you can
Yes,
just
yeah, I'm going to start. So good evening, everybody, and welcome to day 3 of the Humboldt's Day India events. These events are being co-organized by Dr. Robin Vijayan from IISER Tirupati and Professor Uma Ramakrishnan and I, Vivek from the National Center for Biological Sciences, Bangalore. These events are supported by the Museum and Field Stations Facility, NCBS, and the Biodiversity Collective. To take you through the talks this evening, we have Professor Karthik Shankar from the Center for Ecological Sciences, Indian Institute of Science, Bangalore, as moderator. Karthik's research group uses a range of small vertebrates in forest systems, as well as coastal and marine fauna, as model systems to understand evolutionary and biogeographic patterns and assist in conservation prioritization. Over to you, Karthik.
Thanks, Vivek. First, congrats, Umar, Robin, and Vivek on organizing this event. It seems like a great success. It gives me great pleasure to welcome and invite Dr. Walter Jetz for today's first talk. Dr. Jetz is a professor of ecology and evolutionary biology and of forestry and environmental studies at Yale. Walter works on the patterns and processes behind the distribution of species and their traits in space and time. He investigates how environment, ecological, and macroevolutionary mechanisms combine to determine the co-occurrence of species and the structure of assemblages, and then to use this as a basis for assessing the fate of biodiversity and its functions under global change. I first had the pleasure of meeting Walter when he, if he remembers, reviewed and rejected my first macroecology paper. No hard feelings. He signed his review, so I got an opportunity to engage with him, and we met at a conference a few months later, and based on many of his valuable inputs, that paper eventually did get published. So thanks, Walter. But at the time, Walter was already by the time I encountered him, Walter was already a young star in the field. He published some seminal papers in Science and PNAS on continental patterns in birds, and I absolutely do not need a little bio sheet in front of me to list all of Walter's papers that I read by the time we started our first work in macroecology. Walter has gone from strength to strength including macroevolution, to his macroecological work. And I, like the rest of you, look forward greatly to hearing what he's done in recent years. Thanks for joining us, Walter, and over to you.
Thanks, Kartik. Thanks for the kind words and for giving me. And thanks for having me at this, what looks like a really successful set of talks and events. So really wonderful. Congratulations to you. And I love this way of actually being able to engage with you, not just right now, but I think it's a great way to engage with distant place and students over there. So thanks for having me. And let me dive in here. I'm going to share my screen. I hope this works. And my talk today is on the global diversification, conservation of terrestrial vertebrates. As we, I hope all know, appreciate, vertebrates, terrestrial vertebrates, tetrapods are an extremely diverse group covering the whole, almost the whole of the land surface. And there's a lot of them. It's over 30,000 species. And after some 10, 15 years ago, so we were able to advance birds as a system, as a global system, one of the first global systems of this size, magnitude, to study macroecological and macroevolutionary patterns and drivers. We are now able to expand this view and this approach to tetrapods as a global study system, and could argue as a global model system for macroecology and evolution. And here they're very differing patterns of richness. In some cases, yes, quite some similarities among birds and mammals in particular, but then the reptiles, amphibians, showing quite disparate patterns. And obviously I could spend an hour just talking about these patterns, and I would love it, but I would rather actually go on today to talk about the macroevolutionary underpinnings of some of these patterns that we're seeing. And that's been, I think, one of the really exciting developments over the last 10 years or so, that phylogenetics, available sequence data, and new methods have allowed us to look at the global diversity of say, birds, not just spatially, but also in the phylogenetic and macroevolutionary dimension. And here, a first attempt to include every single species of extant species of bird in a tree, with the colors characterizing diversification rate or speciation rate, essentially in red, the parts of the tree where there's been a lot of recent rapid radiation. These are hot parts of the bird tree of life, and cold parts in dark blue. And what was so cool here is that by having every species included, we were able to actually compare every species to every other species in how rapidly radiating its part of its lineage, its part of the tree may have been, or not, for that matter. And we were able to identify some groups that been particularly rapidly radiating, gulls, for example, and white eyes, just another example at the bottom there. And I think you'll hear from Moyle tomorrow, and he's done some really cool work on white eyes. I'm not sure whether he's going to touch on it, but so this is the tree, and then really cold parts here over here, the oil bird. I'll come back to that. But that then allowed us to map species, not just in their aggregate sort of richness, but also in the sort of the average past signal of the speciation rate, diversification rate that they convey in the place where they are today. And we find that it's quite a heterogeneous picture, that there's not a simple latitudinal gradient. But what we find is that there actually, there's actually almost an east-west difference in, and some interesting difference, for example, among islands and mainland, in that islands tend to harbor species that have very short terminal branches that are relatively recently rapidly radiated on average, whereas on mainland that's on average not the case. Their species there tend to be in lineages that are darker on that tree that I showed you earlier. There isn't much of a latitudinal difference, so more rapid radiations, more rapid speciation would be a simple way to explain why there is higher species richness in the tropics for birds. We were then later able to take that into the third sort of elevational dimension here, work by a PhD student Ignacio Quintero, who has looked at yes, obviously the decrease of species richness here on the y-axis, species richness of birds along elevation for various parts of the world, and generally always a decrease towards higher elevations with slight variation in the shape. But what was intriguing here is that by adding that signal from the phylogeny into the picture, by adding that speciation rate, that tip diversification rate, we call it also, that we measure on the tree, so that redness or blueness of the tips that I showed earlier on the tree. By bringing that into the picture, we're able to show while species richness always tends to decrease towards higher elevations, speciation rate, so whether or not these species are red hot or recently rapidly radiated, tends to increase with elevation. So you find at higher elevations more rapidly radiating species groups. What that suggests is that given that we have low diversity at high elevation, yet these are highly rapidly radiating clades that we find up there, it suggests that what's going on there is some sort of ephemeral speciation, so that there's a lot of extinction happening there, high elevations, but then a lot of rapid rediversification, so which makes these lineages up there on average relatively recent, relatively young. Just fascinating. And there is an interesting latitudinal and also other environmental interaction going on here. We find that increase of that higher speciation rate and that increase of the speciation rate towards high elevation, particularly in places that have seen a lot of past rates of temperature change, so that there has been a lot of perturbation of these assemblages over the last hundreds of thousands and millions of years. So it's those places where a lot of extinctions would have happened, we assume, and where then a lot of rapid radiations followed. So that was quite, I think, interesting to be able to add that other dimension, that other phylogenetic perspective to elevational gradients. And here, next group that we looked at, work headed by Nate Upham together with Jake Esselstyn and myself, look at mammals, all mammals of the world. So similar sort of approach that we applied to birds. And the point I'm trying to make here, and that Nate makes very well, is that he makes through this little animation that we obviously didn't just look at a single tree. One of the important advances here and needs was to cascade the uncertainty that the still missing data brings into the tree by working with multiple trees, ultimately a sort of a pseudo-posterior distribution of node ages and of trees, so whole thousands of trees that we ultimately developed, and that all show slightly different patterns. But once you run your analysis over a whole large selection of these trees, you're able to bound the uncertainty, characterize the uncertainty that's still in the tree, and that will always be. There isn't, we're never going to have one perfect true phylogeny for all mammals. So what that work allowed us then here is to look further into macroevolutionary patterns, not just spatial variation, but here we brought in some traits as well. Vagility has been argued a lot to matter for diversification in mammals, and indeed we found earlier suggestions confirmed that less well dispersing, less vagile species of mammals tend to have higher speciation rates. So if you're really widely able to move far places, you're obviously able to connect with other populations more readily, and speciation rate tends to be lower in that case, whereas less fragility promoting allopatric speciation. That's our interpretation. And we also found that diurnality played a really important role in speeding up in some of the diversification in the tree of mammals. So diurnal species carrying a signal of higher speciation rate. The latitude, again, as in birds, essentially no signal there, which is intriguing from a sort of global macroecological perspective, given the high diversity of mammals in the tropics compared to the higher latitudes. So here, a simple spatial look at richness patterns, not actually giving us that full picture. And I think that's been one of the interesting things in macroevolution, in biogeography actually over the last 10 years, that the ability to bring in that signal from the tree of life. And it was kind of an evolution for myself, having started out as a sort of more classical macroecologist. I really felt, now we gotta bring that other dimension in, and that's really been immensely helpful for tetrapods, but also for a whole suite of other species groups. And we'll hear more from Rosie later, Rosie Gillespie, and obviously we already heard from Rory Bowie yesterday. This is, I think, one of the most important areas of biogeography in the last 10 years, that ability to link up macroevolution and macroecology. So with tetrapods, then we now have a pretty good system. We essentially have not just the bird and mammal tree completed, but also first attempts at the squamate and at the amphibian tree, and also the crocodilian and turtle tree. This was also partly made possible through the NSF support for the BirdLife project, and Rory is a collaborator on that, as well as several other folks. Alex Pyron was a key collaborator, is a key collaborator on this work. And with him, actually, we did some more recent work on amphibians, looking at this, at distinctness. But let me start out with birds first, where flipping it a little over now to this conservation aspect, away from a more macroevolutionary process and pattern analysis. Here, putting this diversification rate that I was talking about earlier, or speciation rate, flipping that to the kind of the inverse. Here now in red on the tree, the species that have particularly long branches through the tree, in particular the terminal branch. So essentially, it's the same tree that I showed you earlier. It's an example tree, one of many trees that we produced. And in red now, the species that have been particularly slowly, or lineages that have been particularly slowly diversifying. And what we find here is, yes, it's not the white-eyes and it's not the gulls. Those were the rapid diversifiers I was talking about earlier, but it's the oilbirds, the shoebills, the hammerkops, the ostriches, etc. of the world. And having every species accounted for, every extant species, we're able to compare them directly, and we're able to look at the what essentially is a sort of a summed, apportioned branch length, sort of going from the tip into the tree. Summing that up, we find that the oilbird is at the top of the league here. That may change a little bit depending on which backbone you're using, but it's actually the top 10. I would expect very little variation going forward. Those are the species that have been isolated on the avian tree of life for a long time, been going their own ways. Actually, the terminal branch for oilbirds, it actually goes back into beyond the KT boundary. And we have here species that ultimately carry with them millions of years of distinct evolutionary history. And the bars here obviously represent the uncertainty that's still left in the tree, given that we don't have just a single tree, but a whole history of many trees. So this is interesting because those are particularly intriguing species in that they may carry not just a lot of kind of unique path on the tree of life, and thereby kind of unique adaptations, unique functions potentially accumulated, but we see that at least in the top range of these species, these are very phenotypically distinct species that often carry very unique functions. So we could argue about how much that holds up further in the tree, but it's an interesting other attribute for us to begin to consider, and it's one that we now have for all extant species, thanks to these global phylogenies. And we really are talking about some very, very peculiar species here. Let me just do a little excursion over here to the oilbird, which I have a particular fondness for, where I'll try to get to play this video. Obviously, it's not working. No, probably not going to make it right. Anyway, this oilbird here that I'm releasing in Humboldt's cave in Venezuela what it does is obviously what usually other birds don't do. It doesn't fly out of the cave, it flies into the cave as you release it. And it does so while you kind of hear its clicking sounds, with echolocation navigating its way through the cave. It's the only aerial frugivore, nocturnal frugivore, excuse me, in South America. There are no fruit bats there. It has an extremely particular function in that region as C. dispersus. We did GPS tracking of that species. Look at the home ranges. So here for about 30 individuals, the home range is extending over multiple hundreds of kilometers in diameter. And a really fascinating species that's probably supporting connectivity also and similarity among some of the forests in South America simply by dispersing these seeds over these really, really vast distances. So we are talking about very particular unique species that are popping out on this as these on the tree of life of birds with millions of years of distinct evolutionary history. And that's, I think, a really fruitful field going forward to have a look at, not just in the case of birds, but other tetrapod groups, but I think going further also invertebrate groups and plant groups. It's starting to offer up a really interesting additional dimension with which to look at species macroevolutionarily, but also with view to potential functional and conservation importance. Here for amphibians now, the same exercise, some of the top species there. Mexican spade toad, I think, is the one that look at the 150 million years. So we're talking about double, triple the millions of years that we saw for birds, obviously, because amphibians reach much further back into the past, having begun to radiate a long time before birds. I think place number 3 or 4 there is the purple frog, which was recently rediscovered in southern India, if I'm not mistaken. So here, a particularly valuable species potentially in your neighborhood that needs much more research, as it was just recently rediscovered. But we have genetic data for it, and there it lands, one of the very top spots. We can look at the evolutionary distinctness distribution. Not just globally, where are the sort of how many top, what's sort of the frequency distribution? Are there most species in the middle, or are there a lot that have a very, very high ED? We can do that not just globally, we look at that for different geographic regions of the world, and we find that South America, the Afrotropics, but also Southern Asia hold some of the highest ED species. And intriguingly, these regions differ really in the shape of this frequency distribution, right? So some have their EDs, some regions were sort of concentrated in the middle, rather it's more drawn out. And there's a really interesting kind of further interpretation, I think, that could be taken here in terms of the diversification and historical biogeographic factors underpinning these distributions in different parts of the world. Taking it more over to the applied side, we can look at how distinctness goes together with threat status. And thankfully, at least in birds and mammals, we find that the more threatened species tend not to be the ones that are also very evolutionarily distinct. But in turtles, recently work by Colston et al. And out of Alex Pyron's lab, we found the opposite, unfortunately, that some of the most distinct species are also the ones on the greatest threat of extinction. And another thing that these phylogenies combined with trait data and threat status data allowed was they enabled us to impute, or with phylogeny support, predict threat status for species that haven't been assessed yet. For example, amphibians, over a quarter of amphibian species haven't seen a red list assessment yet. Yet it's an important, powerful tool to get them on the map in many of the policy efforts and conservation efforts. So here, work by Pamela Gonzalez del Riego for amphibians, but there was related work for some of the other groups where we predicted threat status from the data we had from the phylogeny for species that haven't been assessed yet, or that were data deficient in the assessment. And here we find that of the 25% species that were data deficient in amphibians, most of them actually aren't least concern green, but many of them are highly threatened. So over to the red side there. Many are vulnerable or endangered or even critically endangered. So, so this work, the phylogenies, together with other biogeographical data and trait data, allowed us to make these predictions, and they can now be considered more thoroughly in some of these processes. You can also bring together this distinctness metric with other information, for example, of how much species geographic ranges have already been encroached. We did some earlier work on that with birds, and here recent work by Ricky Gums out of London, where he looked at that frequency distribution across different vertebrate groups, and not just evolutionary distinctness, but combined with that encroachment. And we find that that combination of being very distinct and being in a very encroached area is particularly prevalent in species that are already highly endangered here. So the yellow, orange, and right on the right-hand side. You can take these efforts further. A quick check-in with Kartik. Kartik, should I finish up now-ish or at the top of the hour?
Let me look at where we are.
5 minutes.
Yeah, I think we want to leave a little bit of time for questions, Walter. I can see a bunch of questions in the Q&A box.
Excellent. So let me skip through this. Another good set of applications. So I'll try to do the rest in a minute or two. I did want to make the point that if Humboldt was around today, he wouldn't be using these tools. I think he would be excited about these sorts of tools, and he would be an avid data scientist. And satellite data, he would be using informatics infrastructure. Perhaps we could get him excited even about things such as Map of Life, which I'm involved in, and that's trying to bring together some of these tools and modeling approaches to get us to, for example, with vertebrates, again, as the key group, a more higher resolution understanding of species distributions, here kilometer predictions for hummingbirds that allow us to newly identify some of the hotspots of richness and rarity of that group at much higher resolution, and then feed that into conservation decision-making, feed that into efforts such as the half-Earth map, and now the ongoing discussions of the Convention on Biological Diversity on global conservation priorities. Here, a high-resolution priority map for all terrestrial vertebrates, and highlighting some of the parts of India that are not large swaths that are remaining to be conserved, but the ones and in high need, but they're highlighting places that are indeed in high need, but already quite encroached. Here, the human modification index in orange. Those are the places that we do need if we want to sustain all tetrapods into the future. And that's setting up very different challenges for different nations in the world. And this is work that we're now feeding into these post-2020 global biodiversity framework discussions. With some countries challenged to do some restoration actually to sustain some of the species going forward. And here a bit of a look at the economic situation. Happy to follow up on that separately. I think with that, I want to leave it there. Here, ongoing work with Mario Mura, where we ultimately arrive at the conclusion here at the bottom that not only is there differences in the conservation challenges, but also there is differences in the need for future taxonomic activity, systematic activity in terrestrial vertebrates. Here we are attempting to identify the places in greatest need of discovery work. And very uneven among nations. And actually, we predict that for the Indian subcontinent, a lot of reptiles and amphibians yet left to be discovered. Probably no news to you, but it's interesting to put that in comparison to other countries. With that, thank you. And thanks to all the people who've been involved in this work. Thanks.
Thank you, Walter. That was fascinating. I think there's a number of questions. Are you able to see the Q&A box?
Let me get out of the presentation mode and I am able, yes, I'm able to see it.
Yes, so would you like to just take those questions? You'll need to call them out very quickly.
So we can start with the first one, speciation rate versus elevation. Some graphs are in a U shape. I mean, some cases speciation higher at low elevation and decrease at some elevation. And yes, good question. So we found that similarly as the decrease of species richness with elevation, sometimes like this, but also sometimes it goes like that. We also didn't find in all cases a straightforward linear or exponential shape of that speciation rate with elevation. So in some cases, speciation rate would go up like this. In other cases, it would actually go down to the middle and up again. I think it's really intriguing. There's so much work to be done to understand what's going on there. And it may go together with particular habitats, climatic belts that we're ultimately encountering. Remember that obviously the tree line also isn't constant, right? It decreases with elevation as we go to higher latitudes. So you're going to have an interesting interaction there as you go from one biome to the next, further away from the equator. The tree line is going to decrease, and some of the climatic belts along elevation are going to change, and richness isn't always lowest at sort of highest at low elevation. Sometimes it's actually higher at mid elevation. Actually, in India, we see that in some cases in Himalayas. So a lot yet to be addressed there. Then any other questions, Kartik, you'd like me to grab?
I think this one about floral species richness. Does floral species richness decrease with elevation, like fauna?
It generally tends to, but we are not able to do that sort of assessment at the global scale in the way we've been able to do for terrestrial vertebrates. So vertebrates or birds in particular are a very unique group in that we are able to look at all species together and characterize them globally now in many of these trends. Generally, yes, floral species richness decreases, but again, in some parts of the world, you'll find the inverse, where actually the lower elevations may be very dry, deserty, and then you actually go to much richer vegetation as you go higher. The Sky Islands, I think, are we going to have a talk about that? I think somebody's going to talk about Sky Islands, I think, so that would be a classical case where it could be inverse. And this, again, in some of the subtropics like India, it may also be a mid-elevation peak actually for plant diversity, and there's certainly been papers that have been showing that.
Participants, I'm not going to take all of the questions that are here, and some of them are a bit detailed, and Walter actually talked a little bit about speciation rates, so Ashwin, I'm not going to take yours right now. But there's another question that says, which animal group is the most vulnerable and threatened compared to others across, you've looked at, I think there's a fair question given that you talked about all tetrapods. Can you pull out some groups that you think are...
Well, okay, I mean, this is not, in terms of, again and again, among tetrapods, we find that amphibians tend to be the ones that are getting most hammered, right? And they're getting it from all sides. They're ecophysiologically require very particular conditions and often wet, humid conditions. So they're very quickly thereby affected by land use change that may be nearby even and affecting their microclimate. They have small ranges to begin with. In many cases, much on average about a quarter the range size that birds have. And they're sitting in places where there's a lot of change happening, as you know, actually, in Southern Asia would be one of the ground zeros actually for some of the amphibian threats, as well as all the way over to Southeast Asia. So that's the one I would highlight. But if you look actually at some squamate groups, some you find the same thing. Crocodilians and turtles. I encourage you to have a look at a recent paper by Tim Colston. There, and in the case of amphibians, that threat, actually there in particular, that high vulnerability going together also with often very distinct old lineages. So something really for us to think about ethically. Hey, can we let this happen? This many million years of unique evolutionary history just disappear.
So there's several questions more for you, Walter, which you could actually answer in the Q&A box while the other talk is going on.
I'll do that, yes.
But I do want you to very quickly, I think, answer one question about conservation. And I think, if you could just it says, how would the results from these studies help in the half-earth concept for conservation? Is the portion that's proposed to be left for wildlife based on endemism? Could you just like give us a very
very briefly, I encourage you to have a look at the half-earth map, and I put the link up earlier. Just type in half-earth map and in Google, and it will show up. And there we offer up a bit more explanation. So in essence, it is something like endemism, right? So we want to think about the places that harbor particularly geographically restricted species, right? Because if something was to happen there, those species would be gone and lost for forever. So in the half-earth map, we're identifying the most important places for the safeguarding of species. So the places we cannot lose if we don't want to, if we want to sustain, in this case, tetrapods species going forward. What we are used for in practice is a complementarity approach. So we feed in the core occurrence matrix of all tetrapod species, of all 32,000 species, and do a spatial planning exercise, an optimum reserve design exercise, if you will. So if you try to identify the places that give you, for particularly many species, that meeting that conservation target of a certain minimum portion of the range conserved. Now that often goes together with centers of endemism, but not always. So I encourage you to have a look at that in more detail at the half-earth map. Don't want to take more time away from Rosie, so I'm happy to answer any more questions in the chat. Thank you so much for listening, and I'll follow up in the chat box now, Kartik.
Thank you, Walter. Thank you for a great talk, and that was just absolutely delightful. Thank you so much again.
Thank you.
Do we have Rosemary online? Great. Gives me great pleasure to welcome Dr. Rosemary Gillespie for our second talk of the day. She's a professor at the Department of Environmental Science, Policy and Management at the University of California, Berkeley. Her research focuses on understanding evolutionary patterns and processes amongst populations and species, with a primary focus on islands, particularly remote hotspot islands of the Pacific, to gain insights into the nature of processes shaping communities over evolutionary time. Dr. Gillespie has many awards, and she's held many distinguished positions, such as the president of the American Genetics Association and the International Biogeography Society. There's more online about her than I can fill in these few minutes. So I'm going to hand over to her, and I look forward greatly to hearing what you have to talk to us about today, Rosemary. Welcome again.
Well, thank you so much, Kartik. Can I share the screen then? Probably, let's see. And I can, I think, just go, if I can get that here, and if I can go to, so see if this works. Can you see that okay? Do you get, oops, let's see. So maybe, what can you see there? Can you see the screen, or can you?
Uma, you're not seeing it either, right?
No, it's the green, it's the green button at the bottom.
That's sorry, the green, you can't, just a sec. Let's see, so just two shakes. So you couldn't see the share screen, right?
No.
Not yours. We can't see your screen. But at the bottom of the Zoom screen, there's a green.
Right.
Oh,
Yes, we can see. Yeah.
Perfect. Okay, and just two shakes, and okay. So can you see that okay?
Yes.
Yes.
And it shows the slide rather than the presenter view. Perfect. Okay, so what I'm going to do today is follow up kind of from what Walter was talking about, because what was really great to see in Walter's talk was this huge macro scale approach to understanding what is going on in the world, what processes might be going on. And so what I'm going to talk about here is how you can look at this from another perspective, coming kind of from the bottom up. And so what I'm going to do is look at how you can use islands to understand, to really get at processes that might be involved in diversification. And I'm going to try and get at a couple of things here. In particular, what I want to focus on is that the idea that islands are, I mean, they're famous for their isolation and how you can get at certain biogeographic processes. But often what you hear more about is the idiosyncrasies between lineages, and islands are unique for this and that, and they have incredible this and that. What I want to do today is try and tease out the commonalities to try and understand actually what the processes might be that are going on. What I want to do is ask, is there anything predictable about what organisms colonize islands and what might be the subsequent pattern of colonization? Okay, so thinking about islands, what's so special about them? The big thing, of course, is their isolation. And they can be extremely isolated, and even ones that are not isolated have been used for looking at balances between immigration and extinction. But the ones that are supremely isolated are the ones that I've been looking at and trying to understand in that supreme isolation that MacArthur and Wilson talked about in their famous monograph. They said near the outer limit of the dispersal range of a given taxon, speciation and exchange of newly formed autochthonous species within an archipelago can outrun immigration. Basically, what they were saying is that this, first of all, that it's very serendipitous as to what can get to these extremely isolated localities. And then what happens in those localities is that you tend to get adaptive diversification. And so I'm going to talk about those two processes in particular. How do organisms get there? Is it entirely serendipitous? And then secondly, how do they diversify? Is there something predictable in terms of the pattern of diversification? So first of all, how do they get there? And so we'll look at spiders, which is my group, and so we're going to focus on those a little bit when I try and illustrate some of these things. But first of all, you think, well, how do spiders get to these islands? And there are various, if you focus on Hawaii as the most isolated archipelago in the Pacific, and you say, well, how do spiders get to these islands? And you think, well, they could, they could balloon out there. There are many winds that come across the islands. And you think, well, can winds get through the Pacific? And what I want to argue is that actually the winds that are the most important are the storm winds. And this is not a new idea. I mean, Lindsley Gressett argued this many moons ago. The importance of storms and major trajectories in pulling organisms from one locality and dumping them in another. And so this would argue that for wind dispersed organisms, for Hawaii, the source is going to be the Americas. And if you think, well, really, is that true? You don't have to look very far. This is actually the fires, as you see in the last couple of days in California, just a devastating catastrophe. But one thing it has shown is that these are the smoke patterns, and you can see that the smoke is actually almost reaching Hawaii. So it does show you that the trajectories are moving out from the continental Americas to the Pacific. So this would suggest that groups that are wind dispersed are coming from the Americas. And so if you think of spiders, oops. I just want to show here, the spiders put out silk, and what they do is they let the silk out, the wind pulls and pulls and pulls, and then poof, off they go. And this is how spiders tend to disperse. And so you say, well, really, are they related in Hawaii to the Americas? And what we found is, this is just a very basic phylogeny, but here you've got Hawaii in the central Pacific. Where these are Tetragnatha long-jawed spiders. they're related to the Americas. And what we found is there are many groups that of spiders that appear to be related to the Americas. They've come to the Central Pacific from the Americas, and presumably through wind dispersal, which you'd expect. And then, you know, but there are other routes of dispersal, of course, and we've heard of many of these. And for example, there are many, you know, we think of birds carrying organisms across the Pacific. And so you would say that if birds are carrying organisms across the Pacific, you would look at the birds that actually do cross a Y. And here you've got things like the plover, the Pacific plover that you get that migrates right up and down the Pacific. And so what would you expect there? For organisms that are carried on birds, you'd expect a relationship right across the islands of the Pacific. And so what spiders here are carried by birds? The most famous one we know that gets caught up in bird feet are crab spiders. So let's look at crab spiders. What you see in crab spiders is that the ones in Hawaii, they're related to the Society Islands and the Marquesas Islands. So they're related right across the Central Pacific, which is really, you know, a fascinating corollary here as to what we would expect. And finally, you know, this is fairly recent, very tentative work that we've been doing as well in terms of ocean currents. What would you expect if organisms are rafting? What would you, the main thing is that they would go from island to island. So they'd go from small, they'd colonize atolls. They'd jump from atoll to atoll, and they'd necessarily land at the shore, and they diverge up the islands. And what you see was so interesting. This is a barracuda, or a tarantula kind of spider that you get in the Northwest Hawaiian Islands. Its relationships are right across the Central Pacific, Micronesia, New Caledonia. And likewise, you know, even on these little kind of rock-like atolls, likewise with Pacific wolf spiders. And this is, again, very preliminary work with Kaina Privet, who's in France. And so what she's shown is that actually, you know, the Hawaiian the Hawaiian lycosids are related to others across the Central Pacific, New Caledonia, Papua New Guinea, and Australia. And we need to do, of course, more work in the Central Pacific to understand what's going on there. But what seems to be going on is that these organisms that you get at low elevations have colonized right across the Pacific, just in a stepping stone-like fashion. And so you'd expect them to colonize these littoral layers and then adapt up. And so what I'm highlighting here is that you've got a series of expectations. And so as Darwin said, you know, when you get to these islands, then what you might expect is that you diversify once you get there. So we've got a series of expectations then for how organisms get there. And then, so now what happens when they reach the island? And so as Darwin said, the more diversified the descendants from one species become in structure, constitution, and habitat, by so much will they be better enabled to seize on many widely diversified places in the polity of nature. So the point here is that once they reach these very remote islands, whether they're rafting or whether they're ballooning or going by birds, how do they then diversify? And this is what I want to just focus on next. How is diversity generated once they get to these islands? And so what I want to think about here is patterns of speciation. And now we've all thought about, you know, how organisms diversify. And what I want to emphasize here are there are fundamental differences. So we think about adaptive radiation, and we think of the classic Dolph Schluter text on adaptive radiation. And so in that situation, what you find is the situation that Dolph Schluter in his book, he highlighted how organisms might adapt to different environments. And so you get disruptive selection, whereby entities adapt to different environments. And the classic example, of course, there is the sticklebacks that have gone into different glacial lakes, and you get a limnetic and a benthic form. And they repeatedly evolve this benthic and limnetic form. And so the question then for me was, how does this divergent selection and add ecological speciation into different areas, how does that play out in the long term to lead to adaptive radiation? Well, certainly, you know, you look at other islands, and here's a beautiful system of wolf spiders in the genus Hogna in the Galapagos Islands. And probably similar things are going on in the Hawaiian Islands with the wolf spiders. So what Frederick Hendricks and his student Charlotte de Bashir were able to show was that in the Galapagos, these lycosid spiders have colonized each of the islands and repeatedly diverged into high and low elevation species. The high elevation is most closely related to the low elevation. If you look at beetles, he's shown a similar thing in beetles. Highland species are genetically closely related to lowland species, and they've repeatedly evolved this kind of divergence. Likewise, in many plants, you get disruptive selection between environments. And so different plant lineages have repeatedly evolved to similar kind of environments through disruptive selection, pulling them apart. So what you find here is that this pulling apart into different environments allows repeated evolution of sets of taxa adapted to parallel sets of environments. So this is what, you know, these are kind of textbook examples of how the environment provides alternative selection pressures and pulls things apart and initiates divergence. And the question then is this the basis for the adaptive radiations that we see today? And what I would argue is actually there are other things we have to be thinking about. And so what I want to highlight here is work that we have done. And I should say that this is, that many, I've discussed these ideas with many others, but the important thing here is that diversification in the course of adaptive radiation can happen without changing environment. And we can get at this in particular in Hawaii because the islands of Hawaii present a chronosequence. This is just a beautiful system where you can look at the older islands and the younger islands and hence see how diversification plays out across the time period from the oldest island of Kauai to the youngest island of the Big Island. So you can see, you can use the islands as a temporal framework to understand how speciation is happening. So let's look at this a bit more closely. And so what I'm talking about here is that you can get situations theoretically where there's no divergent selection between environments, where chance events dominate the initiation of reproductive isolation, presumably genetic drift in allopatry, founder events, population bottlenecks. And then you get the species coming back together. Now, many have argued that this would take so long that it's just not going to be an important aspect of adaptive radiation. But let's look a little bit more closely at what's going on in Hawaii, given that we have this chronosequence. So what I want to highlight here, this is the work of Anna Hiller, who is a student in my lab. And she did niche modeling across different lineages of Hawaiian arthropods. And what's really nice about this is that it highlights the range of the entire lineage. First, you see this is Drosophila flies, this is Lapala crickets, this is Meso-Sydney plant hoppers, and this is Tetragnatha spiders. What you see is the plant hoppers are shifting niches a lot, But if you look at the spiders, they're largely in the same habitat across the entire lineage. They're largely in wet forest habitats, and they just sit there and diversify. And so let's look and see what happens. What I want to, I'm going to just show you a couple of lineages here. And first of all, I'm going to show you the lineage Ariamnes. Now, these are stick spiders. And what you find is that they have repeatedly evolved similar forms. And so here you get, there's ones that live under rocks that tend to be really dark brown, and ones that live under leaves that tend to be gold. And you get these same forms evolving repeatedly on each island. So parallel sets of species on each of the islands. So if you look at how these evolved, what you see, what I've illustrated here is the purple on the top is the oldest island of Kauai, and then you're going down the island chain, and so it goes purple to turquoise being the youngest island, purple being, I mean dark blue, sorry, being the oldest island. And so what you see is that you get repeated evolution. This is the gold form. This is a white form that lives under lichen. This is a brown form that lives under bark. And so you get repeated evolution of these same forms, so that in any one habitat, you tend to get one representative of each of these species. Now, I'll tell you why I'm highlighting this in a second. You get the same kind of thing in the genus Tetragnatha, and this is the one I'm going to highlight. You get the same sets of species in any one habitat. So this is what I've called the big brown species that you get on bark. And this is a representative on Kauai and on the right Maui. This is a maroon one that you tend to get in mosses. And this is a green one that you get on leaves across the islands. And this is a little brown one that I've called that you get on twigs. So how have they evolved? These are the green, maroon, large brown, small brown ecomorphs. How have they evolved? Across the islands, basically they go from the oldest to the youngest island, which you'd expect, Kauai to Maui, to the big island, just down the island chain with some differences going on there. But importantly, so you get the hopping down the island chain, of course. And the thing is, you get repeated evolution of similar ecomorphs. So here's the big brown morph, the maroon morph, the little brown ecomorph, and then the green one. So repeated evolution of the same form. And what I'm going to focus on here are these green ones that you see here. And this is work that Darko Koutaris, who was a student in my lab, he looked at these green forms that are so closely related and tried to understand what is actually going on the youngest islands to promote diversification. So what he did, so he, we're looking at the green forms. So he looked at the youngest islands, and here you get multiple, it's the different ecomorphs all live happily together. But he was looking at the green ones, which are different shades of green, illustrated here. But basically what we knew coming in was you get one species on one part of Maui, then another species, and they have these kind of parapatric distributions. So Darko wanted to understand exactly what was happening here, and to just see, how do they differentiate all, on the same volcano? Are they just kind of diverging genetically? So what he did was he did some genomic, he did exon capture work, looking at these green species that are just starting to diverge. And what he showed was really quite cool, because you get this species in allopatry as well as being on the same volcano. So what he found, and I'll highlight what's going on here. So here you get the very, very structured populations. And so you see here, this Lanai one is here, and this Brevignatha, this is on the big island that's here. So you see, if we highlight on Lanai, this is a different species actually on Lanai. It's very genetically and morphologically distinct. It's still a green one. It's a green ecomorph. And this one has become a new species. This is on the Big Island. And what's interesting is that they're all also on East Maui. And so what that suggests is something really quite intriguing based on the data that Darko showed. He showed not only do they have parapatric distributions, these three species, on this one volcano, but they're also kind of interdigitated across the volcano. You get patches of one taxon, patches of another, and they don't interbreed. Now, why is that so exciting? The big thing here is that, you know, on the youngest island, it's all the same population. Here you've got the second youngest island where you get interdigitation. And what this is suggesting is that you've got a period of allopatry that's providing genetic isolation. And then you have secondary sympatry. We don't know for sure, but this is what this is indicating, given the temporal framework that we have. It's secondary sympatry that leads to the interaction. And then you get subsequent ecological divergence that allows stable co-occurrence, because at some point, this interaction appears to lead to character divergence. So what this suggests is that these species within these lineages are not diverging into different environments. We know for sure they're not. They're all in wet forests. It's almost the same. I mean, it couldn't be more identical, really, across the islands. So species seem to be diverging entirely through biotic interactions. So they're coming together, close relatives are coming together and diverging, as compared to the environment for classic ecological speciation. And so this situation seems to be the one that allows repeated evolution of sets of interacting taxa. So just kind of comparing these situations, you've seen situations of disruptive selection between environments. And so, for example, in when we were talking about the sticklebacks, this is frequently ephemeral. We know in sticklebacks that you know, they diverge, limnetic and benthic, and they frequently come together. They fuse, the environment changes, and they come together. The isolation is lost, and so this is really well illustrated in the situations of speciation reversal: cichlids, sticklebacks, whitefish, Darwin's finches, even. The situation that I've highlighted in the spiders is geographic speciation. It may be less ephemeral. It doesn't rely on maintained divergent selection imposed by the environment because they're diverging in the same environment. And these situations are more likely to be impacted by invasive species that disrupt the interactions. So what I've tried to highlight here is that only by understanding the differences and the conditions creating the differences between lineages will we ever understand the processes involved in the formation of biodiversity, and hence how it's going to respond to future stressors. And what we're trying to do now then is just understand the evolution of entire communities. We're looking at evolutionary processes in the context of how lineages are changing, as I've been talking about, and putting this together. So individual lineages, how they change through time, and then putting this in the context of ecological interactions. You've got the island chronosequence. You can see communities of different ages. So can you put these two things together? And so understand how entire communities evolve through space and time. And with that, I just want to thank you for having me. And it's been, I've really enjoyed this forum for talking to you. And I should say that this talk was inspired through the work I've been doing with the American Genetics Association and the symposium we had with many people working on adaptive radiation. And I would be delighted to answer any questions that you might have.
Thank you so much, Rosemary. That was fantastic. I think, you know, as students 30 years ago, when we were first introduced to MacArthur and Wilson's theory of island biogeography, what was notably missing from their own framework, which they acknowledged, was a lack of, you know, of their ability to incorporate evolutionary processes in it. And, you know, to see that, in your work 20 years later, and also the fact that it resonates so much with the work that we've done, Robin and Uma and Vivek and all of us in our work in biogeography in India with the islands, that I think it's like really provides wonderful insights. Thank you so much.
Well, thank you, Kartik.
Are you able to see the question and answer?
Yes, I can, and I can't quite see. So let's see. Should I start with Deblina?
That's the one. That's the one that.
Yes. Yes, population of any flora or fauna depends upon wind direction. All of them are the same phylogenetically, or are they different, or can change phylogenetic properties? Now that's that's an excellent question, but yes, I mean there's so what I was trying to highlight there is that there's a set of attributes that seems to go along in terms of the predictability of the direction with which species will come. And yes, what the important thing I think seems to be important here is that the trade winds appear to have little to do with dispersal patterns. They just kind of circulate around. It's really the storm trajectories. And as I said, Gressich was the one that really highlighted this, because he said that storms have the ability to pick up and dump down, whereas trade winds just circulate. And so this is what we seem to be seeing here. And certainly other things that you might expect for taxa like this are, you have to look at the storm trajectories for the given wherever you are in the world. But the other thing is that you're looking for organisms that tend to be dispersed by wind, of course. But the other thing, it's sometimes really hard to tell for spiders in particular, because they use multiple mechanisms. But the other thing is looking at the kind of habitat that they live in, and also what I think is fascinating is really just the expectation of where they would land. And this is something that Karlquist talked about, is that it's much easier to get somewhere than it is to stay there, to establish. So he argued that there was very limitations on dispersal. There were basically no limitations, and he argued that it was establishment that's a real kind of bottleneck. And with wind dispersal, it allows you to get to somewhere that is your habitat. It allows you to have niche conservatism, to maintain your niche conservatism, and get to a place that matches where you started. That was a long answer to the question here. Should I go on to the next question?
It would be nice if you could read it out, because I think the participants can't see the questions.
Okay. So, is being airborne dispersal over thousands of miles really the most plausible explanation for Hawaiian spiders forming closely related to American species? Are there other candidate hypotheses? I think that question, well, so there are absolutely. The other hypotheses are that they could, I mean, there are only a certain number of hypotheses, because, that they're ancient lineages, and, they're 5 million or more years old, and they must have got there somehow. But I think what I've tried to highlight is that even, during the smoke plumes that you see, you can see how trajectories can get over to these islands. And I think perhaps the most important answer to that question actually is that these things are extremely rare, and that's what we need to emphasize, because if there was colonization of Hawaii, like planes arriving there, you would never get the adaptive radiation that you find today. There are very few spider lineages. The ones I highlighted are pretty much it: jumping spiders, long-jawed spiders, wolf spider. I mean, there's just not that many of them. And so how often would have spiders have got there? You know, we say, well, every year? No. Every 100 years? No. Every 1,000? No. I mean, it would have been once every 100,000 more, you know, or you know, even fewer years for that you would have had a successful establishment of the islands. So it just, what I want to emphasize is the extreme rarity of organisms getting out there and allowing them to then radiate. Now, I hope that answered the question there. Should I answer a couple more or one more?
Last one from Justin.
From Justin. Oh, so Justin said, I'm interested in how flyways and fly-throughs in the Pacific Ocean have contributed to extant population structure. How much has been done on this? Very little. I seem to see less literature on these topics. I was also wondering if it would seem that ocean air currents are more likely to diversify or homogenize populations, or perhaps both equally. So these are all fascinating questions. So I think this area is just ripe for research because there has been very little follow-up on these kinds of questions. And I think now with genomic data, we could actually try to, you know, get an understanding. There's some tantalizing bits of information about, you know, how birds carry certain organisms and you know, what gets stuck on feet and looking at gut content and survival through guts. I mean, we've got all the pieces there. So now can we basically retrace the evolutionary history and see, you know, actually where did they arrive on the islands? You would expect the aerially dispersed taxa not to arrive on the shore. You would expect the ones that are rafting to arrive on the shore. And so in terms of homogenizing versus, you know, allowing diversification, this is something, again, it's the balance between the number of propagules you're getting to a site versus the time they have on their own. And what's really interesting there is, you know, if you look across different radiations, I know I didn't talk about this, but what is very clear in many radiations is that the island of Kauai was the most isolated. The island, because when the oldest island of Kauai first formed, it formed at a period when the other islands were low and far apart. And this is work that Jonathan Price and David Clegg did, showing that most of the diversity of Hawaii reached, you know, started on the currently oldest island of Kauai. And what we see is most of the ecological splits happened on the island of Kauai. Many of the organisms that diverged ecologically on Kauai then hopped down the island chain. And so this, I think, highlights your comment, Justin, that that it's when they're supremely isolated, this gives them more opportunity for ecological exploration. But again, this kind of question, it's ripe for looking at, and it'll be a great thing to really study and really get a handle on.
Well, thanks again. You know, it's a shame that we have so little time, because I think there's you know, questions that not just the participants but all of us have for both you and Walter. But I'm going to hand back. But before that, thanks again from all of us to Professor Gillespie and Professor Jetz. Walter and Rosemary, thank you so much again. Lovely talks and very inspiring. I'll hand back to the organizers now.
Well, thank you.
Thanks, Karthik.
Thank you, Karthik.
Thanks very much for this. So we'd like to, of course, thank all the attendees also for coming in and you know, staying on for these talks. So we have again a great set of talks tomorrow, and I hope all of you can be here. And at this time, we'd also like to kind of bring up something that we've started talking about from 2 days back. You know, when we had this India series. And this is about inclusivity and biodiversity in biogeography research. So there have there have been some more comments that have come about this, and we have a small poll. So please do stay back and just vote on whatever you think are things that you can you think are relevant for biogeography researchers. And also, please feel free to use the Q&A section to type in anything additional or anything new, any ideas that you may have, and we'll try and include that in the polls tomorrow. So at the end of these 5 days, we'd probably have a kind of a long list of interesting ideas. Thank you very much. And now over to the polls.
Thanks, Robin.
Thanks, everyone.
Yeah, thank you, Karthik.
Thank you, Karthik.
Thanks, Karthik. We're hoping to get back some insights from these polls to the IBS, International Biogeography Society. Let's see where that goes. Let's see where we can go with this.
Oh, it's a great idea. Great.
So while we are doing the votes, I'd like to bring up this point that the last option in the poll was actually suggested by one of you, like one of the audience yesterday.
Yesterday, yeah.
Kind of added that in today, which is undergraduate lecture series. So yeah.
And actually,
please, if you have any other,
yeah,
do mention the question and answer.
In fact, I just picked that one today.
This is actually something which IBS is considering actively. There's been a lot of discussion about this in recent meetings. So hopefully this is something which will happen.
So I'll go ahead and close the polls in about 20 seconds.
Thanks again, everyone. You've closed, Vivek?
Yes.
Yeah.
Okay.
Thanks, everyone.
Yeah. Bye.
Bye-bye.
Thanks, Rosemary. Thanks, Karthik. Thanks.
Thank you all.
Walter. Thanks, everyone. Bye.
See you all tomorrow.
Nice to see you all.